we'll probably get a thing saying it's recording. Sweet. All right. So welcome everybody. Thanks so much for coming to, this is episode 5 of our ASB Student Author Ask Me Anything series. And today we have Ridhi Sahani, who may or may not be in one of these directions. We've done some on a really macro level of biomechanics lately. So today we're gonna go all the way to the other end, or almost all the way, depending on your length scales, and look at the cellular level. And so Ridhi is here from UVA, and she's gonna discuss her recent publication of diaphragm muscle fibrosis involves changes in collagen organization with mechanical implications for Duchenne muscular dystrophy. And then answer our questions about how she actually did this and other things like that. So thanks so much for everybody for coming, and Ridhi for talking to us, and take it away.
Great, thanks for the intro, and thank you everyone for tuning in today. All right, let me just share my slides. Looking good? All right, awesome. So today I'm excited to talk about my publication and also just general research process and answer any questions you all have. So to get things started, before we jump into the research, I want to talk a little bit about how I got here. So one thing I thought about when I was reflecting on my paper and just the last few years of grad school was that research is really a series of questions and then some answers and then some more questions. So some of my questions that I've tried to answer were, first, what do I want to do with my life? So a little bit of background about me. I was born in New Delhi, India, and immigrated to the US when I was 5 years old, and mainly grew up in Newtown, Connecticut. And so for my undergrad, I went to Bucknell University in Lewisburg, Pennsylvania, where I majored in biomedical engineering and minored in dance. And that really shaped my interest in human movement and biomechanics in general. And so while I was in undergrad, I did some biomechanics research at a pretty large scale. So looking at playground injury prevention and designing assistive devices. And then from there, I knew I was interested in pediatric focused biomechanics. So I applied to a REU program at the Children's Hospital of Philadelphia. And so while I was there, my research focused on automotive safety, looking at child occupant kinematics and autonomous emergency braking. So at this point, I knew I was really interested in biomechanics, but next question was, what do I want to study in grad school? And during that process, I decided that I was really interested in incorporating more biology and physiology into my biomechanics knowledge. And so I joined the multi-scale muscle mechanophysiology lab at the University of Virginia to study muscle and pediatric neuromuscular disorders. And so with that, then the next big question was, what do I actually want to research? So when I joined the M3 lab, a major project that was ongoing was looking at Duchenne muscular dystrophy, or DMD. And so for anyone that's not familiar with this disease, it's a fatal and very devastating muscle wasting disease that impacts skeletal muscle across multiple scales. So it occurs due to a lack in expression of the dystrophin protein, which sits at the muscle cell membrane and is responsible for the mechanical integrity. And so in the absence of dystrophin, every time there's muscle contraction, this leads to more damage and a chronic state of inflammation, which occurs due to a buildup of extracellular matrix, or ECM components. And so if we look at a cross-section of skeletal muscle, in healthy muscle, we see this nice uniform structure, but in DMD, we see an infiltration of fat and fibrosis and variation in muscle fiber sizes, which all reduces the force generating capacity of muscle. And so in these patients with DMD, degeneration progresses with age and use, and eventually leads them to require the use of a wheelchair in their early to mid-teens. And so when I got to the M3 lab, I thought all of this was super fascinating. I felt like this was something I could be motivated to study for as long as a PhD would end up taking. But the big question was, where would I fit into this project? And so the previous models that had recently come out of the lab had highlighted how complex fibrosis was in DMD. So Kelly Virgilio was a grad student who had just graduated, and she built these micromechanical models at the muscle fascicle scale and found that the ECM properties themselves really influence the macroscopic tissue level properties. And then another model that she developed was an agent-based model, and it revealed the importance of the density of the ECM on muscle regeneration. And in these models, the effects of collagen within the ECM were really just based on its amount. And so at the same time that I joined the lab, another student who is now graduated, Hunter Wallace, was doing experiments to characterize the role of collagen amount on diaphragm tissue properties. And so in DMD, a lot of the field has focused on the lower limb previously, but the diaphragm muscle contributes to respiratory insufficiency, which is actually a leading cause of death. And so our lab was sort of shifting to focus on the diaphragm muscle as well. So when I joined, we started thinking about beyond collagen amount, what's going on with its organizational structure. So started thinking about in the diaphragm muscle, what's really going on with the collagen. And so for a little bit more background, diaphragm muscle is essential for respiration. And so when during inspiration, the diaphragm muscle contracts and shifts downwards as lungs fill with air. And then during expiration, the diaphragm muscle relaxes as air is pushed out of the lungs. And so in DMD, we see a weakening in the strength of the diaphragm muscle. And so if you look at the vertical distance of the diaphragm muscle between inspiration and expiration, it's a marker of its efficiency. And so these are from a patient with DMD. And as you can see with age, there's a decline in this diaphragm muscle excursion. And so in order to study a diaphragm muscle weakening, a really common animal model that's used in our lab, as well as many others, is the MDX mouse. And they actually replicate the degeneration seen in patients in DMD quite well in the diaphragm muscle. So we also see a reduction in the strength and elasticity of the diaphragm muscle tissue and an increase in collagen density. However, the increased amount of collagen does not correlate with the increased tissue stiffness that we see in the diaphragm muscle. And oftentimes we quantify fibrosis with increased levels of collagen. But really the collagen is organized quite complexly in the ECM. And so the skeletal muscle ECM also has these different hierarchical structures. So the epimysium surrounds the whole skeletal muscle and is shown to have unique arrangements across different types of muscles. And then the perimysium and endomysium surround individual muscle fibers and muscle fascicles, and they have this honeycomb structure that's seen across different types of skeletal muscle, and actually increase in these collagen cables is reported with fibrosis. And so often when we characterize fibrosis and skeletal muscle ECM, we see muscle cross-sectional images, which capture this perimysium and endomysium organization, while the epimysium remains more largely uncharacterized. And so that's where my study focused. And so the first question when we started this study was how collagen fibers are organized in the diaphragm muscle. So we really weren't sure what would be going on and if we'd see any changes with DMD. And so to do that, I had to start with taking a closer look and doing some imaging. And so to do this, we first dissected samples of diaphragm muscle tissue and then digested the muscle fibers out to isolate the ECM. So this was done with a sodium hydroxide-based digestion. And then imaged in the surface plane using scanning electron microscopy to characterize the collagen fibers. And now this all nicely fits on one slide, but I would say this probably took about 8 months or maybe a little bit longer than that to really get nice looking images that we could then investigate further. So with that, we started looking at healthy and MDX mice from 3 to 12 months, and qualitatively, as you can see in these images, we see some differences in organization. And so the next question was, how can we quantify these changes that we see in the diaphragm muscle tissue? And so to do that, I developed some image analysis methods. So first, we measured the muscle fiber direction. So as I mentioned in these samples, we digested the muscle fibers, but we could see these ridges corresponding to their locations. And so we measured those and then cropped and rotated our images such that the muscle fiber direction aligned with the horizontal axes to keep this consistent. And then we also measured the collagen straightness parameter manually in image J. And this was done by outlining the length of a collagen fiber and then a straight line connecting the ends of the measured fiber. So with this measurement, a value closer to 1 signifies more straight collagen fibers and lower than 1 is more wavy fibers. And then the next thing was developing a way to characterize the local collagen fiber directions. And so I did this in MATLAB by first discretizing the image into local image windows. Next, the image was binarized, and from this we can measure the collagen pixel ratio, or the amount of pixels associated with collagen in that region. And then from there, we used canny detection to outline the fiber boundaries. And then at the fiber boundaries, we computed the radon transform, which it computes the line integral across a series of angles. And so then the degree at which the peak intensity of the radon transform occurs at is the degree of maximum alignment. And so we can use that as the local collagen direction, for example, shown here to be 72 degrees in this image. And so this was measured relative to the muscle fiber direction in all of the local image windows. And so then overall with this algorithm, we can input our SEM images and then output the local collagen direction per image window. And then this allowed us to look at both the mean collagen direction as well as the strength and alignment. And so the strength and alignment is also a value between 0 and 1, and a higher strength of alignment signifies that the fibers are more aligned in that mean direction. And so with that, the next question was if we see any differences quantitatively in organization with age and disease. So the first thing we saw was that the collagen pixel ratio did not show any significant differences in our groups. And also the collagen direction was consistent between the groups as well, but was between 70 to 90 degrees. This was actually pretty surprising because going into the study, we didn't know that the collagen would be aligned in that transverse direction. So this was pretty interesting to find this and see that it was consistent between all of the samples as well. And then some of the more exciting results too were that the collagen fibers were straighter and more highly aligned in the MDX mice and the older healthy mice. And so we saw at the 3 and 6 month age, especially, that there was an increase in straightness and alignment in the collagen in the MDX mice. And so at that point, we kind of thought we could end this study here. We have some interesting imaging results. We see these changes in organization, but really the big question was, do these actually influence tissue properties and how could we predict its effects? Also being in the M3 lab, we really love modeling. So if there's an opportunity to incorporate that, it's probably something you're going to end up doing. So with the next question, I was interested in how to predict how these changes that we see are occurring affect tissue properties. So to do that, needed to use some more math. So overall, the way that I went about this was using the SEM images and then the local collagen directions to develop a finite element model. And so for these FE models, the size corresponds to the region of tissue that was imaged in the SEM images. So it's about a 120 by 120 micron region. And for the FE models, each element corresponded to one local image window from the SEM image. And so for these models, we used a coupled solid mixture constitutive model to represent the ECM. And in this model, collagen was represented with a toe linear fiber material. And so collagen regulates the tensile properties of the ECM. And with this, it only produced force in tension. And then the remaining components of the ECM were represented with a neo-Hookean residual ground matrix material. And for this analysis, we kept all of the material properties the same, and the only thing we varied were the material axes. So again, these were assigned per element based on the image processing. And then with these models, we simulated a 20% equibiaxial stretch to measure effective stiffness. And so we did this by using assigning the boundary conditions. And then as you can see here, we can see a dispersion in stresses across all of the elements. And then from that, we calculated the average element stress and average element strain to measure an effective stiffness at 20% strain. Sorry. And so doing this, we saw that looking at the FE model outputs, we see some qualitative differences in the dispersion of stresses. And so shown here in the MDX images and models on the top, you see a higher region of these transverse stresses. And then also for both of these, higher transverse than longitudinal stresses as well. And then the next question was to see how these changes in organization affect the properties in different groups. And so first we saw that the longitudinal stiffness was increased in the wild type mice. So again, at those 3 and 6 month time points, the wild type models predicted an increase in longitudinal stiffness, while the MDX models predicted an increase in transverse stiffness, which was again the direction in which those collagen fibers were aligned. And then the next thing we did from having this pipeline was comparing the organization measurements with the tissue stiffnesses. And so interestingly, the collagen fiber direction didn't correlate with the predicted tissue stiffnesses, but both the straightness and alignment did. So as we see an increase in straightness and alignment, the models predicted an increase in transverse stiffness, and with a decrease in fiber alignment and straightness, there was a decrease in an increase in longitudinal stiffness. And so after going about this series of questions and some answers, this really boiled down to two goals of the study. The first being looking at changes in collagen organization, and this was done using SEM images. So some of the main findings were that the collagen fibers were oriented transverse to muscle and increased in straightness and alignment with fibrosis. And then the second goal was to use in silico methods to predict the influence of these changes in organization. And we found that collagen contributed to the transverse properties and that changes in collagen organization were implicated on these, and that there was a positive relationship between fiber straightness and alignment with transverse stiffness. So with all that, I also wanted to talk about some things that I learned through this publication process. So like this Bitmoji, definitely had a lot of times of feeling lost along the way, but I would say one thing that I'd recommend is definitely start drafting early, even before you think you have a complete story. It's okay to just jot your notes down as you go and break down sections. I found starting with the methods to be really helpful. And then also storyboarding or using outlines are a great tool to keep you organized. Also organize your code and shared resources periodically. So for this paper, all of our code was uploaded onto GitHub, and we used Figshare and other repositories. And so to do that, it will help you in the long run if you keep it organized rather than waiting till the end to get everything together. And then also preprint servers are a great way to get your work out sooner. So I preprinted this paper on bioarchives, which was really helpful for sharing it with people and also attaching it with like fellowship applications and stuff like that. Also, writing groups I found to be really helpful, trying out like the Pomodoro method or some other variations where you take specified breaks so you don't get too lost in the weeds and end up like that. And then also revisions do make your paper stronger. So also just to be transparent, I guess this paper was initially submitted in April of 21 and published pretty recently in March of 2022. So it was a long process, but I will definitely say looking back at the first revision, I think it definitely does make the paper stronger. So some lessons that I learned were that one, patience is hard, but very important. Don't let perfect be the enemy of complete. So there will be a point when you do have to just submit the paper. It will never be perfect, but that is okay. And there's always more questions. So I think another important thing is to decide, are these questions maybe your next paper or something that you're going to add in here, and not be afraid of like pushing it a little further than you can. And so just like collagen, the research journey can be quite wavy. So this is just a little graphic that I put together to describe the publication process, which I'm sure a lot of us relate to. So thanks everyone for listening, and I'm happy to take any questions about the paper or research and publication in general.
Ooh, great job.
Thank you.
Thank you so much. And then, yeah, as Ridhi alluded to, it's question time. So if anybody's got some questions for Ridhi, like how do you get those Bitmojis onto a PowerPoint?
The Bitmoji app is quite helpful.
That works on a PC too.
I think so.
There's a Chrome extension for it.
Oh yeah.
I think Xavier might have had a question.
I do. It's a quick one. Were there any suggestions from the reviewers that maybe didn't change anything about this paper, but made you consider things for future projects?
Yeah, that's a good question. I think yes would be the answer. It was stuff that we were already thinking about, but I think like kind of brought it up to the surface. So one question that they had and that we'd been thinking about was, so this whole study, we're only imaging that top layer of the ECM. So a big question is how much that affects the bulk tissue properties and what's going on with the collagen inside the ECM. So in those endo and perimesial layers. So that was something I was planning to look at next, but definitely was like even more motivated to do that after getting reviewer comments.
Great presentation.
Oh, sorry. Go ahead. Go ahead.
Oh. You mentioned that it took about 8 months to set up your protocol for imaging.
Mm-hmm.
Can you talk more about like from the inception of the initial idea to like getting to the point where you were writing the paper? What was your timeline? Like how long did portions take and what ended up taking the longest?
Mm-hmm.
I guess what would you say the like hardest hurdle to overcome was?
Yeah, that's a good question. I think the longest part was probably like getting all of the images. So yeah, it was a lot of troubleshooting in the beginning, but then also like collecting all of the samples for the different ages. And that was probably the biggest hurdle. But even with the modeling, I think it sort of like seems like this very obvious like framework now when I talk about it, but it took a pretty long time for us to think about like, okay, how do we develop a finite element model based on these images? How do we even analyze them? So there was definitely some back and forth also between like, once I started modeling, re-looking at how I was analyzing my images and like making sure that this was this nice closed loop system. So yeah, if that answers your question, that was probably like another hurdle. And then in terms of timeline, I'd say I started drafting while I was still like running models and refining that pipeline there too. So the drafting process also took like a good amount of like probably another 6-ish months.
Thank you.
Mhm.
I've got lots of questions. I don't know. I guess I'll just do one that's just sort of general. You mentioned that you put it on a preprint server.
Mhm.
Was that something that would you say that was your idea, your advisor's idea? I have an advisor who's kind of anti-preprint, so I'm just curious to see like if you had to convince your advisor or if they were open to it or how that process went.
Yeah, I think my advisor suggested it actually. So I was applying, it's quite a fun timeline. So I was applying for a grant that was due like the beginning of April, and we wanted to get the paper out before that so that we could show it was submitted and then also get it uploaded to a preprint server so that by the time the reviewers were looking at it, they had something to look at, which really pushed the timeline for the paper a bit. But that was really the motivation to put it on a preprint server. And then looking back, seeing how long that timeline was, I think that was also nice.
Sure. Yeah, seems like a huge benefit for us grad students and early career folk is getting, stuff out there.
Definitely. And I will say we didn't like upload any of the like shared code or any of that on the preprint server. So I had links on there, but they were all like embargoed until the paper would be accepted. So none of that was really like public, if that is a hesitancy for
Sure.
that.
Yeah.
Yeah, that makes sense. And so like the idea is out there, but nobody's downloading the data set and doing something else until it's been peer reviewed and everybody agrees it's good to go. That makes sense. Cool.
Oh, actually, that kind of reminds me of a thing. Hi, I just wanted to say, love your slides. They look great. It's very clear, though I do totally understand the kind of problem with getting data in the sense that like 8 months of work gets boiled down into just one figure. For your paper, when you went to submit it, was there anything, I'm trying to go through this process myself right now, stuff that you weren't able to include in the paper, figures, data sets, other things that might not have been able to like make it into the final draft that would have been helpful to put in there? How did you go through the whole process of basically selecting what stuff you wanted to show and how to show it?
Yeah, that's a great question. I would say one of the big things was the figure limit. So I think I had like 3 or 4 supplemental figures, which I think most journals will allow that. So we used like a I think Figshare to upload links to all of those shared figures. So yeah, initially like deciding what are the key figures, what are the ones that can be supplemental. So a lot of the like sensitivity analyses and validation of our methods ended up being supplemental. So that was one thing that I think as I was drafting, I was aware of the figure limit, but tried to just like write out everything and include it. And then when we got to the end, sort of comb through like what was really important to keep in the main manuscript.
Okay, cool. Thanks.
I guess sort of along with that, did your figures like sort of evolve? Because I mean, I know some of the papers I read is like, those might have been separate, but now they're A, B, C, D, E, F, and that counts as one.
We had, I guess from, yeah, that was definitely like one tool for shrinking down the number of figures was the subpanels for sure. It's a great trick. But then also the figures did change during the revisions process. So the way like initially we analyzed it was I think only presenting the ratio of the longitudinal to transverse stiffness and focusing more on that. And then that had raised some more questions. So throughout that revision process too, the way we presented those figures did change a bit.
Cool. But yeah, I'm with Chris, 8 months of work down to one figure. He summed up something I'm about to start working on real quick.
Yeah. Yeah, and it was like a methods figure too, not even like a results figure.
Yeah, but hey, one of our previous AMA people was all about those methods figures. He thinks those are really important. So
yeah. And then they're great because you can use those when you're presenting on your work too.
True.
Oh, I might have missed this. Just in terms of the methods they use for this, I'm just kind of curious because I've been doing some like material modeling stuff. How did you measure like the stiffness of the tissue samples that y'all were getting?
Yeah, so these were just like the model predictions themselves were not like validated against measurements of tissue stiffness. But so for the material, the collagen fiber stiffness, I varied over a range reported in the literature, which was pretty large range. But we did a series of sensitivity analyses looking at how the collagen fiber stiffness itself affected the trends in the data. And so there was basically in our plot a region at which that effect stabilized. And so we stuck within that region of ranges in the literature so that the predictions themselves aren't that sensitive to the material properties, really the orientation of those fibers and the organization.
Okay, thanks. Yeah, sorry, I must have missed that.
Yeah, no, I don't think I went too much into that, but we did add a supplemental figure about this.
Cool. And then speaking of supplements, we have a question in the chat that was from Xavier, is asking that you said that the validation of your methods ended up as supplementary material. Did you consider writing that as the validation as a separate paper?
I actually don't think we ever thought about that, but that is a good question. Yeah, no, we did not.
I have seen some of the sensitivity analysis. That seems to be a way that people are getting modeling parts published that used to like you couldn't get the paper published of it doing something because those reviewers would be mad that it hadn't been validated. And you couldn't get the validation published because you weren't using it to do anything. And so it looks in some of the modeling stuff that I've done on a different scale, that sensitivity analysis that you did seemed to be a way in.
Yeah, the other thing was like the predictions from the model are like the model itself is hard to replicate experimentally. So in the discussion section, a lot of it did focus on comparing to other measurements from the literature because there's nothing that's like an exact replica of this model, which is really why we chose to do a model rather than experimentally measure these properties. So yeah, that's definitely something with models that comes up a lot.
I think also in some of those agent-based ones, they do it a lot.
Mhm.
right? Because that's already a statistics premise. And then
Right.
I think of, oh, go ahead, Danny. Danny might have another.
Oh, yeah, no problem. Just asking about, I guess, kind of the logistics. Did you have, I didn't see what journal this was published in, but I was just curious if you, like some people, it seems like they start out and say, like, oh, this is the project we're going to do, and we're going to try to submit it to X. Or, you know, afterwards, what was the thought process behind like choosing that? Was it, you know, because some are open access or some have a figure limit, like you mentioned, some have a word count? What was that? How did that go, I guess?
Yeah, no, that's a great question. I didn't really start thinking about what journal until the point of like starting to draft the paper. So like as we were going about the study, that wasn't really anything that was at the forefront of my mind. And I think the biggest decision factor for me was looking at like papers that I was citing or reading a lot of what journals those were in. And so I did have like a list of journals, and I ended up publishing in the Journal of Applied Physiology. And the other thing, since this was like experimental and mechanics, was seeing like if we wanted to go for a journal sort of in the middle there or more of a like computational or experimental journal. So it made sense to go with applied physiology. And then another thing I think I talked to my advisor about was like the next steps of the project, like what journal that would probably be in, and trying to diversify that. So that would probably be a more mechanicsy journal. So trying to have a variety. I think that's another important thing to think about.
Cool, that's awesome. Does applied physiology have a word count?
No.
Yeah, nice. Okay.
Yeah, I'm pretty long-winded in my
Sure.
of things which worked out well.
I'm writing my first paper for one that doesn't have a word count right
Oh,
now,
nice.
and I'm just like, I can just keep going and going. And
Yeah, it's a little
Which-
Put
dangerous.
everything in it.
Yes, exactly. Yeah. What if we added this? What if we added this? Yeah.
Yeah.
Kind of dangerous. Yep.
Yep.
I have the opposite. I think jobs right now, Journal of Biomechanics is like 3,000. So like you're done.
Yeah.
Like
Yep.
6 pages maybe.
Yeah.
But-
No.
It can be nice because you can just be super succinct and kind of, I don't know, I wouldn't say leave out details, but you can kind of be selective. Whereas, I don't know, you can't really hide behind that excuse without a word count.
Right.
I think certainly to the reviewers' points, you'd have harder time saying, well, we ran out of space. That's in the
Yep.
next one.
Yeah,
Mhm.
Like-
exactly.
Yeah, in that sense, you couldn't hide. I don't like it when people try to hide. Sometimes when I read papers, I'm like, I see what you did there. But
Yeah.
I guess it got through review. So,
Yeah.
but
Yeah, I would also say the like responding to reviewers part was not what I don't know. I feel like I wasn't quite prepared for that. So just knowing like how you actually write a response to reviewers and what all to include, and making sure you're using the right language and acknowledging all of their comments, even when you think you did. But you know, it's still like if they brought up something, it means it really wasn't clear. So it may have been clear to you, but isn't to another expert.
yeah.
Did you get any questions from reviewers that you had to go back and either recollect data or rerun simulations to kind of address?
Yeah, so I actually reran all of the models. So there were questions about the material properties. So we did reevaluate the sensitivity analysis, and we picked a higher stiffness. So initially, our models weren't sensitive to that value, but it wasn't in the range of literature values. So when we reran that, none of the findings really changed. The values themselves did, but I did rerun all of the models. The images, luckily, I didn't have to repeat. There were some questions about like explaining why we did things a certain way. So I collected additional images on other samples, and I think some of those ended up as supplemental figures or just figures for the reviewers. So luckily, I didn't have to recollect that data, which definitely would have taken a lot more time, especially working with animals and waiting for them to age. So the models were a lot easier to really just like rerun and plug and chug.
Okay, yeah, no, that's my biggest fear is just we're like looking at a thing and they're like, I'm just anticipating a comment being like, well, if you think that this would make it better, why didn't you just do that? And it's like,
Yeah,
'cause that's hard.
right. And I will say with the reviewer feedback, we did, like I talked to my advisor about being strategic, like where can we really address this comment easily, and like making sure to do all of that. And then where can, so like on an experiment, where you like know it's going to take like another year, that might be a place where you try to, like, I don't know, kind of stand up for the way you did it more than just like listening to their feedback and changing that part. So sort of balancing how you respond to different comments.
And it sort of sounds like maybe more of the stuff that they wanted rerun has more to do with the method, right? And making sure that the method is as sound as possible, and sort of less about, well, if you think that would make it better, like, are you going to do that next or something maybe?
Yeah, it was almost, oh, sorry.
No, you're good. I was just going to say some of the reviews that I've been a part of are like, if they have to rerun all the models, it's because like the premise was not quite there or something, or there was a missing link that we just wanted to make sure everybody was on the same page about. Unless like they had stuff they were going to do next as well. And so like that's especially if you phrase it at the end or in the discussion of like, and we think we should do this. I haven't seen reviewers jump on and go, well, you have words left to do it now. Because they know, I guess, right, that you're going to do stuff next. But.
Right. Yeah, that's true. Yeah, it was more of, I guess, the comment was something that looking back on it was kind of like, oh yeah, like, why didn't we just pick that number and use it for our models like that after doing our, like, that made sense. So there were definitely some things where I was like, oh, yeah, that makes total sense.
Just thinking about like kind of the comment you just said, like hearing some feedback from reviewers like that you might not have thought of. Did you, I guess I don't know how many like co-authors and whatnot you had on the paper, but like, did you ever solicit feedback? I guess in the preprint you could do that, but did you ever solicit feedback from like people around the lab or anything like that to try to get extra eyes on it before you submitted it?
So I did have two co-authors. They had like looked through the paper and given some feedback as well. I do think I had a few like lab meetings. One thing we liked to do in the lab, especially there was a certain time period where we all did some sort of like a writing goal for the semester. But I presented like a storyboard of the paper, which a lot of us do in like the early stages, which is super helpful for just saying like, this is how I'm planning to present my data. Does this make sense? Especially to someone who's a little bit outside of this specific area. So that was one place where I got feedback. I think I also sent my paper out to my lab before one of my lab meeting presentations and got feedback that way too. So I didn't really go beyond our lab, but definitely had like different eyes on the paper.
Sure. Yeah. I worry about that because I eat, sleep, and breathe like this stuff. So I just worry that, somebody else is going to be like, I have no idea what you're talking about here. Yeah, that sounds like a good strategy.
I think if you don't lose your family or friends that are not doing PhDs in the sort of intro and then the very last conclusion, then you're like off to a good start. They might not get all this stuff in the middle. You might get lucky and somebody might be really good at grammar. But I've noticed that even just sending it out to people that like, they know it because they know you, but other than that, they don't know it at all. And that's an interesting one to get as well.
Yeah, that's too, especially with like grammar things.
Is that most engineers?
I'm definitely, I struggle.
It's like a formula. I feel like engineers, we should figure it out, but I'm so bad.
Yeah. It's just, I feel like I write the way I think, which sometimes is like very, very long run-on sentences. But like when I read it, it makes sense, just not to others.
Yeah. No, that's great. I think, and then, plug for student chapters, because, right, then you have a couple people that and it's going to be a low stress, hopefully, group, because there's no big PIs staring at it, but you have people that are doing biomechanics, excited about biomechanics, but not in your lab, right? And might come from sort of fields nearby. And that maybe that would be a good way too, to like see like which journals do those people write in, like if it makes a lot of sense to them, or if they see ones that relate or something.
Yeah, definitely. That's a good idea. Add it to the.
There you go. Got the self-promotion in there. My job's done. I had a question sort of about about one of your findings. I was trying to think about it as we were going through the end. I was like, wait a second, that's really cool. And so, because I don't spend all that much time thinking about muscle really in depth and how it works and stuff. And I'm really glad that other people do. And so when you said that it increases in stiffness, especially in the transverse direction, so the extracellular matrix around it increases in stiffness, and you're seeing correlations with that happening when it gets really straight as well. Do you think that has anything to do with like that toe region disappearing? Like it's getting stiffer, so it doesn't do the spring thing that it's like supposed to do?
Yeah, no, that's a great question. So yes, I think that it probably has more of effect on like the length of that toe region. So it's really affecting like when the diaphragm muscle starts to produce passive force. And so the collagen is really like responsible for producing force only when it's fully stretched out. So that was one of the things with these samples, the way that we image them. Oh, bye, thank you. We don't know where the samples sit on that muscle length region because they were like kept free floating in fluid, but it's really imaged at a stress-free state. And so if we compare that 1-to-1, you see that it's more aligned in the MDX ones. And so that would probably mean that in vivo, they're starting to hit that linear region of the passive force curve.
Yes, it's like it's already stretched out. So if that's what's giving it yes, the motion or like the muscle is also shortening, sure. But like if that's all that stuff around it is sort of what's restricting and containing how far the muscle as a unit could go. So then if it's already stretched out, it's got nowhere else to go.
Exactly.
Yeah.
And then if you think about the diaphragm muscle too, so it's pretty unique from other skeletal muscles where the fibers are like aligned in a predominant direction, that it is like this circular thing. So these images are at a pretty small region, but if you assume that that is consistent throughout the entire diaphragm, that potentially means that the diaphragm, like the collagen fibers run in this circular order. So circular arrangement. So it's really affecting the
the
properties.
Ability to expand that circle. Wow. Everything I learned about DMD, it just gets worse. Like, I mean, like physiologically is very interesting and cool, but like people have this and I mean, I know it's devastating but like there's so much to it. So that sounds like there's a research track there if professor is in your future. Which is good for sure. No, that's really cool. Yeah. So I admit I know Ridhi, so I've asked her questions before. I guess some other people here do too. I can also, I guess I didn't announce this at the beginning, I can stop the recording if anybody has questions that they didn't want to ask on the recording. So I'll go ahead and do that now. Bye, YouTube.
Thank you.